So I hope it's better now. So good morning. Good morning to Barcelona. Good morning to this EPPN Symposium. First, before starting, so I have the pleasure to introduce you to some of the EPPN activities we've done. But first, I would start with housekeeping information. So for the post presentations, so as you know, we were a little bit delayed. So the posters can be set up in a coffee break afterwards. There will be marks on the board. So please just ask for in this organizing team for where to put your poster, and please do it in a coffee break or later on. So thanks a lot. The other question or the other announcement would be that we have a professional recording team here to record all the sessions and put them on the website. I hope you don't mind and you agree that your presentations will be presented also to the wider community beyond the audience here. And with that, I would like to start with showing you briefly the activities of EPPN. So EPPN, it's a research infrastructure project. For research infrastructure in particular, I would like to start with the EU, what the EU thinks about research infrastructure, what are the major goals in research infrastructure. So research infrastructure is supposed to be the key element to advance knowledge, technology. It is supposed to bring different stakeholders together, and I hope it works well, that we have a very diverse community here. Then an important thing of research infrastructure is the use of the research infrastructure, enabling access to different users to use the infrastructure. It's also supposed to attract young scientists to do work with this infrastructure, but also to shape the community. And finally, it's supposed to address societal challenges. And societal challenges, when we think about plants, so plants are right in the center of major challenges related to global change, to energy, natural resources, food and feed for a growing population. So it's actually pretty straightforward to address global challenges when thinking about plants. Specifically when plant phenotyping, so plant phenotyping plays a key role in many different disciplines. So quantitative assessment of structure and function of plants is essential for breeding, for functional genomic. It's essential to increase productivity of plants in a field, in an agricultural scenario, but also in a horticultural production. Biodiversity or addressing global change is also pretty important in terms of phenotyping. However, as you know, phenotyping, for instance, the cost for genotyping, for sequences, they are continuously decreasing, and they are decreasing faster than actually the costs for computing. Whereas the costs for phenotyping, probably to phenotype a single plant, they are more or less constant. I don't know of any numbers, but I would suppose it's more or less constant. So there is an increasing bottleneck, and it's important to address this bottleneck to enable basic progress in basic plant sciences and in breeding, which also provides some opportunities, novel opportunities. In particular, having this interdisciplinary field with plant biologists, engineers, scientists coming from many different disciplines. What we need to do is to establish mechanistic, high throughput and field phenotyping platforms and in the end also allow access or enable access to all those platforms for a wide, diverse community. And this is actually where EPPN fits pretty well in. So EPPN, it's a research infrastructure project, which as already addressed, it was started in 2012 and it will end by the end of this year. And the basic structure, there are three key elements in EPPN. The major key element, the basis, it's the transnational access. So transnational access, it means access to different users to the EPPN platforms. There is a lot of joint research activities on sensors, good phenotyping practice with a goal to improve the functionality, operationability of those different platforms. And there is a lot of different networking opportunities with different user communities, from developers, from academic users, from industrial users, which we did within the last couple of years. And I would like to briefly show you some of the results, some of the work we've done within the last years. So joint research activities, what we've done, sensor development, good phenotyping practice, where it means we try to develop standards in plant phenotyping, experimental standards, but also IT standards. I will not go into more detail here. There is an entire session on standards, but you also find a lot of posters about those joint research activities here. I would like to talk a little bit more about networking activities. So what we did within this networking activities, we had many different activities such as workshop with different users, with developers, across Europe in many different countries. We had summer schools, training schools, roundtable meetings to discuss specific topics related to plant phenotyping. We organized conferences, and we were present at many different sessions, at many different conferences where we could address a different community, not only this plant biology community, but also we went, for instance, to image analysis community and tried to address many different fields. The other thing, what we did, so thanks for visiting our website. So we had over 500,000 of visitors on the website, and visitors in this sense means at least 2 pages were visited during one visit, which is pretty substantial. So thanks a lot for visiting our website. I think you have internet access now, so please feel free to visit it again. So I hope we get maybe 100,000 within the last quarter of the year. So the other thing is EPPN. So this is how we started. Those are the EPPN partners. It's 14 different partners. We had also a partner from Australia. The plant phenotyping community, at this time, it was up and running, but it was still pretty small. When you look at the development right now, it's pretty substantial. There is a lot of different activities all over the world, and I'm pretty sure that this is not complete. But I try to summarize the activities where EPPN to a certain extent was involved, or I do not want to claim that EPPN initiated all of those activities, but EPPN played a role in enabling these activities. The other point, so transnational access, what we did, so transnational access, it was the key element in this entire project. So what does it mean, transnational access? We had 23 different installations which were open to the user community for access based on a simple selection procedure, application, review process of these applications, and then the selected users, they had access to the platforms and could do the experiments they proposed. You see that we have 23 different platforms distributed across Europe, and here are probably many transnational access users sitting in an audience, and they will also share their experience with you. How did it work? So we had quite a number of applications, so the demand is higher than the amount of access we could offer. Actually, it would be even higher. There was even more requests from outside of Europe, for instance. And it worked pretty well, and there is still some final activities which will be finalized this year. So the experiments we addressed were extremely diverse, so the platforms are very diverse, and we did experiments on many different plant species, addressing many different topics from molecular level up to canopy level. We addressed also mechanistic understanding of very detailed mechanistic processes on plant level, on cellular level, sometimes up to screening mapping, large mapping populations. So it's a very diverse field. As a summary, the most frequently addressed topics, it was drought, nutrients, and roots. Just few topics. It was, of course, much more. The yellow lines, which you can see here, they indicate the idea of transnational access. So transnational access means you are not allowed to do your experiment in your own country, but you need to cross the border, go to a platform outside of your own country, and do the experiments. It works also not in a way that it's just a service. You propose an experiment and we do it. On the contrary, you are supposed also to be involved in this experiment. So many people went to a different country, stayed with a platform scientist, did an experiment. So the idea is that you learn. You not only do an experiment, but you also learn plant phenotyping techniques, and which you can do together with the phenotyping experts. it worked actually pretty well. We attracted a lot of young scientists, so 40% of the scientists doing the experiments going to the platforms were under 35, a third between 36 and 45, and just a quarter over 46. The gender aspects, it's perfect. And what we also did, many new users. So we did not have only existing partnerships or existing partners would do the experiments with us, but we learned actually a lot of new partners and established new collaborations, new partnerships. The outcome, it's also actually pretty good. So we had 30 peer-reviewed publications. You can also find them on the website. And there was also a special issue which was also linked to the activities of EPPN. So right now 30 publications. I know quite well that there is still a lot of experiments are not ongoing, but the analysis is ongoing. So there will be, we can expect 10, 15, maybe even 20 different publications. So in terms of a 4-year project, which a relatively small budget, it's a pretty substantial outcome. And briefly in summary, so what we did, so there is a lot of development in technology standards, which was pretty important for the operation of those different platforms. Networking, so we did a pretty good networking job by attracting many different people, in particular young scientists, young people, and in the end, the other thing, transnational access, we also practically interacted with different scientists, which is also to a certain extent also a networking activity on a practical level. So in summary, EPPN was a pretty good, or was a nucleus to integrate the plant phenotyping community, and I think it was not only it's a European project, but we also managed to attract or extend the discussion about plant phenotyping beyond Europe. And I would like to acknowledge also a few people. There is a lot of people sitting here in the audience. First of all, the team around José Luis, the local organizers. I do not want to mention many names, but Uli Schur, he introduced himself. He was the coordinator of the project and initiated the entire project. There are many different partners sitting here from the project, which unfortunately I cannot mention any names because it's many different partners who are involved, who put a lot of effort into this work. I would like to particularly acknowledge people who worked in the platforms, so people who did the day-by-day job, who are involved in this running those platforms. So the success of running these platforms determines also the success of this transnational access. So it's not only the scientists leading the platforms, but also the scientists or people involved in a day-to-day business in running the platforms are essential for the success of projects like EPPN. And by that, I would like to briefly also explain you the structure of the project, of the symposium. So what we try to do is to present the work which we've done within EPPN and mostly transnational access. So it's work, you can see it as plant phenotyping was used as a tool to address many different questions by many different users, and I hope you will see that there is a large diversity of questions which were addressed by many diverse users doing quite some impressive work. And the sessions, they look like this. So we addressed most questions, by far the most questions, we addressed abiotic stress. So basically the first day, the entire day would be dedicated to abiotic stress. And on the second day, there will be root phenotyping, model plant phenotyping, but also standards in phenotyping. As you can see, there are some gaps in this entire structure. There are some topics which are not covered, and we try to cover these topics with key lectures. So there will be 2 key lectures on field phenotyping and breeding to give you a more complete pictures about plant phenotyping. And in the end, the project ends, and we would be happy to celebrate with you a little, to slow down, go to a dinner with you, invite you to a dinner, celebrate the achievements of EPPN, and maybe also discuss future achievements. So you are welcome to go to the dinner. Directions how to get there is on your badges. On the back side of the badges there is a small map with the directions how to get there. You can pull it out and it's so you are welcome to go there. The dinner is at 20:30. And by that I would like to finish, and in case you have any questions, urgent questions, please ask now. But I suppose we are pretty late, so I would propose that we continue, and I would like to pass over to Imre Vass, who will chair the next session, and we will start with the next session. So Imre, the